Thanks everyone. Thank you for the invitation to speak today. This is the first time I've spoke really about this work, and it's the first time I've really analysed this work only in the past couple of months, so it's hot off the press. It's not anywhere yet. They're kind of the first ideas around the research proposal that was centred around will climate change worsen the problem of antibiotic resistance. So if it sounds like I don't understand any of the graphs I'm trying to present, it's because I probably don't. So any thoughts or comments are much appreciated. All the work today, especially the experimental work, was done by my postdoc at the moment, Dr. Zoltan Erdos. Without him, I wouldn't have any of these slides. The One Health approach to AMR, which we all kind of know, is the idea that everything can link together, right? So we need healthy humans and healthy animals in a healthy environment, and they all mix together, and AMR can move between these things, and bacteria can, or individual genes, and we all interact with everything, and we need to understand it all to be able to predict how antibiotic resistance will change in the future and how it's changing now. And there's relatively broad acceptance that antibiotic resistance can be selected for and spread in the wider environment in this bit here. But there's quite a lot of argument about how important that is relative to agriculture and humans. And I'm not going to deal with that today. I'm going to take it as gospel that the AMR and the environment is this important aspect that can move between these arrows. And to understand the risks of AMR in the environment, it's a really big interdisciplinary problem. As we've heard in the first talk and the second and third talks today, it's a really big problem that I'm going to come at from a kind of reductionist angle later on. But in theory, you've got people and you've got the environment and you've got arrows that go bidirectionally. And how things can move between these is we can use environments differently, and that could differ the uptake between the two. Different environments can have different selection for AMR, different sources of AMR, and us ourselves can have different levels of AMR, and all of these things can kind of interact to change the kind of weight and size of this arrow and which way it's going and how much AMR is in here. And climate change is going to impact all of those, right? So it's going to change how much AMR we have, it's going to change how we use environments. And what I'm interested in here and during this talk and generally in my research as a kind of climate change evolutionary ecologist is how is climate change actively going to change the selection of AMR in the environment? And is that going to differ across environments, and can we predict and understand and generalize how this might be happening? And we've probably all seen some of these papers recently. It definitely inspired my fellowship applications a few years ago, was that there have been a few pieces now that have taken existing data sets on genomes or resistance phenotypes, basically large data sets, and they've basically done analyses to correlate levels of resistance with environmental variables and population geographic variables. And this very famous paper by McFadden in 2018 found that hotter states on average, well, it was actually minimum environmental temperature that was the correlate that was, I think, was significant. But basically they found that hotter states had higher levels of resistance in hospital settings. And then they used that space for time substitution to say that climate change could make levels of resistance worse, and that would be really bad. And then McFadden, with another set of team, did one for Europe and found similar things. They found that hotter places generally have more resistance. That's since been done in China, and very recently it's been done for the whole world. And all of these things are taking really large-scale data approaches to approaching this problem. And that's really important, it's really needed, but it's not getting us to the mechanism, right? And I'm sure I don't have to remind you all that correlation does not equal causation. And to do that, we really need experiments. We need, alongside these things, alongside these large scale things, we need experiments to understand phenotypes and how phenotypes are changing across these environmental variables that are going to change in a warmer world. And basically these correlative approaches mean that there's so much that we don't know. So we have no idea what is causing levels of resistance to increase as temperatures increase. And here are some kind of mainly ecological or evolutionary possible mechanisms that I could come up with when I spent 5 minutes making this slide. So for example, the costs of resistance, the cost of carrying the plasmid or the resistance gene could be less at higher temperatures. Antibiotics could have larger impacts and therefore increase selection at higher temperatures in the environment. It could favor more clinically important genes. We could have a higher likelihood of compensatory adaptation, and horizontal gene transfer could be higher, and pathogens could survive better. But basically, we don't really know. There's little bits of work on some of these, but compared to like trait-based ecology and climate change ecology, we're really scraping the surface in terms of the phenotyping that we're doing. And these knowledge gaps limit our ability to mitigate AMR, probably because they might have different control measures if we find out which ones are most important. And this is kind of linked to a couple of the talks today. There's increasing need for phenotypes, right? Lots and lots of machine learning are coming out that are using existing databases of phenotypes that allow you to then put in your favorite genomes, and you can get out traits, and you can get out some traits like minimum inhibitory concentration. We really want to be able to predict evolution, the optimum temperature of any given bacteria from just their genome, all of these traits that theoretically we don't need to phenotype anymore. But we also know that existing data is biased in terms of the taxa, but also very much in terms of the environmental conditions that we're testing them in. I mean, up until recently, we were only interested, or we thought that the majority of AMR selection occurs in humans and in other animals. So we were testing all of our MIC assays and all of our phenotypes for pathogens, mainly at 37 degrees, right? And now if we think there's a paradigm shift and we think that selection is occurring in the environment, then how likely is it that we can just take an MIC or a prediction of what the selection is under 37 degrees and say, actually at 10 degrees in a soil in Cornwall, is that level of selection the same if that same amount of antibiotic is present there? And in reality, we just don't know. Those data sets don't exist. And even some of the data sets that do exist are not, I would argue, are not the level of resolution that's kind of needed to be able to do predictions, because lots of these traits change non-linearly, and fitting non-linear curves to be able to predict to new novel conditions is difficult, and we need very high resolution data sets for this. So AMR selection is a balance between being susceptible and being resistant, basically. So in conditions where it's better to be resistant, if you're in red, selection for AMR is likely to occur. And we're basically interested in whether climate change is going to change this balance. So I've basically taken all the questions that I had on the previous slide in the black box and put them to either side of whether it mainly impacts the resistant bacteria or the susceptible bacteria. And one that sits in the middle is whether horizontal gene transfer is higher at higher temperatures. And basically, I'm interested in a few of these questions, and I'll present some kind of preliminary data on this today. So I'm interested in whether horizontal gene transfer will increase at higher temperatures. I'm interested in whether antibiotics have a larger impact or a different impact at different temperatures, and whether the cost of carrying resistance is kind of lower across different temperatures. And I'm not just interested in temperatures, I'm interested in actually an organism's representation of that temperature. So how does it do at a given temperature? And what this means, it allows us to be much more predictive in how novel temperatures or novel conditions will change selection and biological rates of the organism, instead of just looking at point estimates of temperature change from say 10 to 14 to 18, when in reality we have no idea how the bacteria or any organism is doing under those conditions. So this whole project's kind of trying to understand how temperature alters the selection for antibiotic resistance. We're taking a reductionist approach where we're testing pure mechanisms of how temperature and antibiotics change bacterial growth rates. And kind of with temperature, what we generally see is organisms kind of increase their performance up to an optima and then decline. And this is kind of a classic dose response curve where your rates decline with antibiotic concentrations. And instead of, I'm interested in both the interactions to these, but this can get very big and very hard. You're kind of combining two nonlinear models here. So the approach people generally take is to look at traits of interest, right? So instead of considering the whole curve, we'll look at where they grow best, their optimum temperature, their maximum rate, and let's say the temperature which they conk out their thermal maximum. And the same goes for dose response curves. We concentrate on things like the MIC, which technically this isn't an MIC, but it will be the kind of rate at which the concentration at which the rate is 95% decrease from the maximum up here. And then you've got this thing that some of us call the MSC, which is like the minimum selection concentration, which would be when it's when there's but I'll call it the no significant effect concentration today. And that's basically the concentration at which the rate changes significantly from when there are no antibiotics. So that's kind of when do we first see an impact. And this is used a lot in ecotoxicology. And I took some of the modeling approaches from that have come out in the last couple of years. So to test this, all of this is kind of the start and a kind of start to where we're going. And now most of this is going to be Zoltan's work here. So we did E. coli experiments where we had a wild type E. coli. We had that same E. coli with a plasmid here in R1 plasmid that encodes resistance for kanamycin. And because we're doing plasmid transfer assays, we created a mutant that basically allows us to look for transconjugates. And then what we did is we measured dose response curves across 11 concentrations to kanamycin, the antibiotic for which the R1 plasmid is resistant. And we did this across 12 different temperatures. And we also measured plasmid transfer in the absence of antibiotics. So we measured how well this plasmid transfers to our mutant across the same temperatures. And this then allows us to fit these these nonlinear curves to see how they interact and kind of how temperature alters the dose response curve of kanamycin in this single bacteria. So the specific questions we're asking are, how does temperature alter the cost of plasmid carriage? How does temperature alter the response of susceptible bacteria to kanamycin? And how does selection to kanamycin change with temperature, which is specifically the differences in response to susceptible and resistant bacteria? And then we're going to look at plasmid transfer. But importantly, just to reiterate, it's not just looking at the impact of temperature, but how do all of these traits change with the thermal performance curve of E. coli. So we're not just looking at absolute temperature changes, we're trying to look at whether we can predict, or there are patterns between the thermal performance of E. coli and the response we see in these traits. And this will allow us to, in theory, later on with more data, create a more predictive framework from the ground up. So we kind of did a lot of lab work that I'm going to skip through, looking at how far I am today. So Zoltan did lots of lab work where we basically set up plates which were kanamycin concentration on one, different strains down rows, and these went into plate readers across 10 temperatures, and across those same temperatures, I think I said 12, across 12 temperatures, and across those same temperatures we then did plasmid transfer assays. And this was very data hungry, it gave loads and loads of data, and we did a lot of data processing to try and clean that up. And we basically estimated growth weight per well. So this is an example plate that we have here. And you can see we get lots of these S curves that kind of link to logistical growth. And we basically grabbed the growth rates per well as our metric of how well the bacteria is doing under those conditions. And then we did analysis. So the curves that you'll see and the data you see is kind of all been analyzed. I think they're still going on, but most of it was done over Christmas when we had some time to do and get into some models. So I'll just show kind of an outline of what we've found so far. So the first thing you're going to see is some thermal performance curves that were taken in the absence of kanamycin. So no antibiotics here, and you see temperatures go from about 16 to 46, and each panel is going to be a different strain. So I'll just explain this one first. So you're a wild type, you see that we see a perfect, very classic unimodal response curve. So the bacteria is happiest at around 40 degrees. and it's barely growing at 16. I think this ran for 4 days to get the full curve in a plate reader, because they grow really slowly at 16 degrees compared to less than a day up at the top here. And then you can see by 46, it is no longer very happy and is start conking out. And we didn't go higher than that because Fisher told us we would void our warranties for the machines if we went to 49 degrees. And then if you look at Naledixic and wild type, you see very similar curves. So actually, the presence of the plasmid and the presence of this kind of mutation for naledixic acid resistance isn't changing the thermal performance curve of the bacterial strain very much. And basically, they all have optimum temperatures, like plus or minus 0.5 degrees at around 41 degrees centigrade. So the things are happiest at 41 from the analyses we've done, and they are not very good at 16, and beyond 46 they're really unhappy and starting to really suffer. And basically I want you to take this 41 degrees, because in reality I'm going to compare lots of the other things to whether selection and costs of resistance and plasmid transfer also fastest or most or greatest at optimum temperature of the thermal performances. So we can look at that through an impact of temperature on response to kanamycin. So these are our classic dose response curves that we've modeled. So I've just shown you the one at the temperature closest to optimum here. And again, red is resistant and blue is susceptible. And you can see that the red, so the resistant bacteria maintains resistance across all of the concentrations of kanamycin that we have, from 0 micrograms per mil to 60. So that resistance doesn't change across the concentrations we're doing. However, the wild type dies before it gets to 20 micrograms per mil, right? So it can no longer grow at any concentration above 20 micrograms at 40 degrees. And each panel is a different temperature. So I'm going to unveil them all now, and they basically mainly show the same thing. So across all of these, you can see that the red curve, the red line is straight and doesn't change. And that means that kanamycin, the resistance gene is maintained and maintains resistance across all of these concentrations. And the kind of shifting up and down of the curve at low temperatures, so at 16 degrees, this is the temperature effect, right? So this is the fact that they grow worse at lower temperatures. And basically, I'm interested, and we're interested in whether the shapes of these dose response curves are changing across temperatures. And maybe you can see a little bit, but it's very difficult, right? It's a complex problem to analyze these nonlinear curves and kind of compare them, and your eyes can play tricks on you a little bit. So instead, what we do, as I explained earlier, is we're going to pick out a few traits, and we'll see how those change across temperatures. So the traits of interest here are the triangle, which is the kind of the concentration at which we first see an effect on growth. You see EC50, which is the concentration at which your growth goes down by 50%, and our estimate of MIC, which is when you stop really growing at all. So do these concentrations change fundamentally across temperatures? And that's what we're going to show here. So this blue dot is the optimum temperature of growth that we found earlier. And basically, this is going to be the effective concentrations. And basically, if you're higher up on this graph, it means that if you're susceptible, you have a higher tolerance to kanamycin at that given temperature, which we would take to mean lower selection. So if we show these, again, just to reiterate, the squares are your MIC, your circles are your 50% reduction, and your NSEC is your when does it first change. And basically you can see a few things here is that basically kanamycin is having a really big impact at very low temperatures here and very high temperatures beyond the optimum. And that actually the kind of concentration at which we first see an effect seems to be less sensitive to temperature compared to the concentrations where we see larger effects, such as the EC50 and the MIC. And actually the level of selection we see isn't obviously linked to the optimum temperature for growth, which I'm still not completely sure what to make of that or what we can take from this meaning. And obviously it's only a single strain. And actually the highest tolerance to kanamycin is occurring at these intermediate temperatures between 30 and 36 degrees. A different way to kind of look at the same data is to actually quantify selection, right? So at any given temperature and kanamycin concentration, the relative fitness of the wild type is the wild type growth rate divided by the growth rate of the plasmid. Okay, and when there's no antibiotics, this allows us to quantify a cost of resistance, because you'd expect the plasmid to be costly, right? So you'd expect the relative fitness of the wild type to be above 1. So you'd think that the wild type should have higher growth up here compared to lower growth down here. And actually, as this arrow goes down, if you're down here, then there's a lot of selection for resistance. And if you're up here, there's not much selection, and that plasmid is costly. So in the absence of any antibiotics, we really didn't measure a plasmid cost at all. All of these kind of credible intervals over cross 1, which means that there's no significant difference between the growth rates of the wild type or the plasmid carrier. But what this does allow us to do is look at how selection for resistance changes across temperatures. So, for example, at 20 micrograms per mil, which is going to be in dark red, that I'll add in a second, you can see that 20 micrograms per mil is beyond the MIC of all of the curves across all of the temperatures, and you can see that there'll be strong selection across all temperatures at 20 micrograms of kanamycin. And just for here, I've put on the human, kind of the internal temperature of a human and the internal temperature of a chicken, which is only 4 degrees, about 36.5, 37 to 41. But we'll see here that even that 4 degrees can really change selection. So the next ones I show are 10 micrograms and 5 micrograms. And you can see that as soon as we're predicting under a bit lower levels of kanamycin, you can see that selection's really temperature dependent. So at 10 micrograms, you can see that it rises up to about 32, 33, and then, sorry, gets less, that's selection getting less, and then selection really increases down to really strong selection again at high temperatures. And this could mean that actually our predictions of MIC that we would originally take at 37 degrees may not be good enough, or the level of selection might not be, we shouldn't necessarily be completely confident in if we are then applying that to other environments. And finally, I'll cover how plasmid transfer alters, how temperature alters the plasmid transfer in the absence of kanamycin. And again, I'm putting on the optimum temperature for plasmid, for optimum temperature for growth of the bacteria. In reality, we were expecting these to marry up, right? Because lots of people think plasmid transfer rates linked to the growth rates of the bacteria. The more bacteria there are, the more they're bumping around. Maybe the more plasmids are going to transfer to each other just because they're in contact a lot more. So when we tested this, we do indeed see a unimodal relationship. We see that plasmid transfer is really hard to measure at 16 degrees and at temperatures beyond the optima. But we actually see the optimum temperature for plasmid transfer to be at a little bit lower at 36 degrees. But this plasmid transfer is really considerably changing across the temperature range. And these are going from 16 degrees that you might see in the environment all the way up to 37 and 40 degrees that you'd see inside a human. And basically, nearly no one's ever measured this over a sort of environmental variable in this resolution before. So we're hoping that this is useful going forwards to kind of understand kind of these fundamental phenotypic parameters that are going on. So just in conclusion, we saw that the MIC can change by more than 4 times across the thermal tolerance of E. coli or this specific strain, and that selection and plasmid transfer changes with temperature in our strain of that we used. Selection and plasmid transfer is not highest at intermediate temperatures, and it's highest at intermediate temperatures, not at the optimum temperature for growth. And I think what's this hit home for me is that prediction of AMR in the environment is probably going to be really hard. And in answer to the question that I gave at the beginning, I really have no idea whether I really think climate warming will worsen AMR from an ecological and evolutionary perspective. And we really need more data in this high resolution on more strains and more species and antibiotics, probably even on resistance genes, because the cost of resistance is likely to change across environments based on the gene, to really look for generalities. But I do think the framework that we've used is going to be useful going forwards to really understand these genotype by environment interactions. And I really enjoy being here because it means I can maybe make some more collaborations, which are going to be key between disciplines. And finally, I just want to say thanks to more people that I can really mention, specifically Zoltan, who's a postdoc who's driven this walk forward. Angus is my long-term mentor, who without which I probably wouldn't be in academia right now. April Hayes, we've been discussing some of these ideas more generally, and Tatiana Dimitri started a URF up at St. Andrews. She is doing a lot of work on plasmids and helped us understand plasmid transfer assays as people who've never done them before. And finally, a shout out to all the open source software developers, without which very few of us would actually do any of the analyses that we do. And specifically, I've put some of the packages that were key in the analyses I showed you today. And they're my conclusions again. If anyone has any questions on them, I'll leave them up.